the oxidating products are produced during digestion in the stomach and intestine, and the antioxidants present in the food. And here we were focusing on the best antioxidant in nature, in food, which is wine. And that was able to prevent two things very important for us. One, a change in lipoproteins in thermooxidative terms, and two, an activation of inflammatory reactions in the colon. And this could be of interest to us. So, in practice, within the body, either we are above 10 micromolars, and we never are, or measuring the antioxidant capabilities in the body just doesn't make sense. So here we are with our shopping bag, full of wonderful substances that chemical labs have told us work as antioxidants, and we have to think about what they do. Now, to do that, I sought inspiration from a significant concept of David Sinclair, published last year in Cell, which I think can give us an understanding, an interesting view. And it's this, the concept of xenohormesis. Basically, these are his words. He says that heterotrophs, that is animals and fungi, are able to sense chemical cues synthesized by plants or other autotrophs in response to stress. Very simply. So if we look for certain protective substances like wine, for example, that are present in wine, the old farmer used to know it, that wine has protective substances. He didn't call them protective, he called them something else. But when the vine has suffered, when there were parasites or drought, activating a protective mechanism, because the physiological response of the fight or flight that's typical of animals, well, plants can't fight or fly. If they have a problem, they have to remain where they are and try to deal with it. And they evolved a defense mechanism that is hugely more refined than ours. We've learnt to use vitamins from plants, but perhaps we can learn more than just using vitamins. And in fact, if you consider polyphenols and other phytochemicals produced in plants that deal with aggression, according to xenohormesis, these compounds could in fact somehow protect us, the animals who've eaten those plants. It's a very complex concept, but in an effort to kind of sum up what we've seen happening to many phytochemical products or natural products like in plants, well, we can see that it all makes a lot of sense. In fact, we can see interference of natural polyphenols with the cell cycle, platelet activation, interference with signal transduction pathways, with very important survival genes via activation mechanisms of kinase substances, with an effect that is anti-apoptotic at low doses and apoptotic at high doses. The plant can do what we do. When there is a certain type of damage, apoptosis occurs. It tries to repair. If it can't repair, it activates a caspase, non-caspase apoptosis mechanism. And certainly, natural products will also combat inflammation. Another interesting area is the control of lipoxygenase within inflammatory reactions. So, a coffee bean in which mechanisms are activated of physical and chemical damage in a grain of coffee, you have lipoxygenase activation. And the lipoxygenase is controlled by polyphenols that are present in that coffee bean. And how much or how little antioxidant exists will determine how much the coffee is oxidized and how good the coffee is. So it's the same mechanism as what happens when cell stress is activated. And recently, we ourselves published a work in Neurone or neurons under stress, and the lipoxygenase reaction has to be controlled by polyphenols to do exactly what happens to the coffee bean. So in reality, we're describing a set of functions, polyphenol functions in plants, where there is a modulation of the stress response. And promotion of survival. The plant has to survive somehow. Since it can't fly, escape, this is how it does it, which opens up an interesting topic relevant to this conference. And here I'm mentioning Medizov, who looked at a very general picture of the causes and consequences of inflammation as an element of abnormal response. Something a bit like what I said before happens in the coffee bean. In the coffee bean, you have a response, but if it's too much of a response, the coffee is bad. If it's not enough, it's also not bad. There has to be a response. And again, what is inflammation for? Inflammation serves to combat infection, to deal with metabolic stress, or and the inflammation occurs, it's also able to respond to the infecting agents. It can repair tissue and adapt to stress. If inflammation goes wrong and is not well adjusted, and there is too much inflammation, in each of these situations you will have damage caused. Typically sepsis, or inflammation, or autoimmunity, which means the defense is wrong and we're over-defending until we may die due to collateral damage. On the right-hand side, you've got abnormal homeostasis, metabolic syndrome, and that's what's relevant to our topic today. If we alter the inflammatory response, then we can have metaplasia, fibrosis, and conditions that oncologists call tumor progression. So, in other words, we're defining the actual time when the activity of natural substances control stress response and optimize it so that it's not excessive. Therefore, biochemists always like talking about metabolic syndrome, cancerogenesis, etc. And at the end of the day, the common soil is fairly, as I said, common. So I'll try and go a little bit faster through my last slides. I'd now like to talk about antioxidants, non-free radical scavenger antioxidants. Just a couple of words to clarify the terminology. People always talk about free radicals, antioxidants, accumulations of free radicals, etc. Now, there are certain molecules that do not work because they block free radicals, but because they shift nucleophiles. One of these molecules is NC-cysteine, but there are a number of other nucleophiles that are very important in the diet, and we'll hear more about this later on. Now, these substances, in fact, here we've spoken about this in biochemistry. They are involved in signaling the signaling functions of reactive oxygen species. Hydrogen peroxide can cause damage, for example, but it can also cause signal translation. And molecules that react to hydrogen peroxide are antioxidant but not free radical scavengers, and molecules that are sensors and translators of hydrogen peroxide sensors may modulate the signaling redox. So there is a signaling redox that is begun by hydrogen peroxide molecules and controlled by natural substances. So, in summary, low levels of hydroperoxides, for example, those in wound healing substances, embryogenesis, or in certain cases of controlled inflammation, lead to proliferation and cell growth. Typically, this is the pro-oxidant condition that some call oxidative stress, but in actual fact, it's a physiology supported by a shift towards oxidation of certain signaling pathways